the third session. In the third part, I'm going to discuss about some methods that are mainly invasive. They are really they're really inspiring methods, but in real clinical practice, at least in the field of psychiatry, there are still kind of challenges how these methods are going to be useful. But I mean, definitely learned about brain kind of in details with some of these really important methods. So I'm going to start with a kind of a study that two Nobel kind of winners, they have done that in 1968, Hubble and Weasel. And what they have done, basically, they have recorded neurons in the occipital cortex, in the visual cortex of cat, and they showed that there are neurons that are specifically sensitive to direction. So these are the firing. So they do neural firing when we have things in a specific direction. If the stimuli is another direction, they would not make that firing. So that is how they have done these studies. If you just look into YouTube, you can find their videos. I highly recommend you to see those videos that they show cats different stimuli, and the cat that specific neuron that they are recording from is not responsive. But when they show that a specific stimuli in a specific direction, then the neuron would start to just fire and make the noise, and they can record that. So that's a kind of interesting idea. So they started from 1968, and they got the Nobel Prize in 1981, if I am correct. So, and it's quite interesting that how they have been successful to do that. Okay, what they do, or what we do right now, we have a specific electrodes that we can put inside the kind of live human brain and record in different levels. So in each point of these electrodes, we can record. So all of these points are recordable points. So we go into the brain, and as you can see here, this is something like 1/5 of millimeter. So it is really, so they are so sharp and so tiny in a way that when they go inside the brain, they do not make significant amount of injury. They just go be around the neurons and go down, and then they record from each of these electrodes that are really close to each other. So 50 micrometer is almost nothing. So when we talk about neurons, that's almost the size of kind of a large neuronal cell body. So they can record that, and there are different kind of details and technology for that, how they are doing that. They call them polytrode instead of just electrode, because they are not really electrode. There are hundreds of electrodes in one kind of line, so they call them polytrodes. And the interesting thing about polytrodes is that because they are recording from different electrodes, so different electrodes could record from different neurons, and then we can make a map. So this is one probably neuron, this is another neuron 3, 4, 5, 6, 7, 8. So we can see that we can even localize different neurons in the space based on the information that we are recording from these electrodes. Okay, and now there are some ideas that we probably can even differentiate the type of chemical materials that they have inside these neurons based on the type of signal that we are recording from these neurons. Okay, and there are some kind of groups of people that are doing that in terms of even not only kind of differentiating these neurons from each other, but also say that, okay, this is a dopaminergic neuron, this is a glutamatergic neuron, this is an adrenergic neuron. So even differentiating them in terms of the chemical that they have inside. But the challenge for these methods is, at the end of the day, we need to put these electrodes inside the brain and record. There are some figures in this chapter I'm not going to discuss about, and I think that They are sometimes too much in terms of the details that are not going to be helpful for you, but for most of you that who are basically beginners in the field. So probably you do not need to go to the details of these things. But there are some specific softwares that are recording from these polytrodes and do the analysis. Most of the time, data analysis for these polytrodes are going to be really complex. We have different types of these polytrodes, so we can put them in different parts of the brain. Some of them are kind of more sparse, some of them are more condensed. We have them in different shapes. And at the end of the day, what people do, they open the skull, they put these electrodes over the head. Some of them are recording from the top of the cortex, some of them are going to the deep part of the cortex, and they start to record. And then they just put a level of sort of the plastic cast, getting sure that these kind of areas in the brain would not be in touch with outside and there would not be an infection. But it's a really complex process to do. And most of the studies that we have with these methods are just people who were supposed to be undergone a surgery for, especially seizure surgery. So when they have a specific area, they need to record. So most of the time they use these kind of, we call them clinical grids. So they use them for clinical applications because they want to remove one part of the brain, for example, this part, and they want to get sure that this part of the brain is not very critically involved in a specific cognitive function. But other than that, they add another kind of research grid to the brain to be able to record things that are not related to that specific disorder. And that's the only way that they can do research with these methods. And this is going to be really complex. So there are very few centers across the world that they have the quality to be able to do these studies. But the good thing about what they can do, they can measure what we call evoked potentials, okay? So they can give people a stimuli, an audio stimuli, and then record what is happening in trial 1. So they give them these audio stimuli one time. So several times they can hear the stimuli. And then if they make an average from them, it is called auditory evoked potential. So this is the average from all these signals that we have. It's called auditory evoked potential. And then they record that auditory evoked potential, and they measure specific maps that we call the power spectrum. These are going to be too much, I think, for those who are especially in the beginning phase of learning about cognitive neuroscience. I just brought these figures to tell you that they are too much for beginners. But just to give you an idea, this is what we have in terms of the time. This dimension is time, and this dimension is frequency. So we have in each time point, for each frequency, we have the amount of energy that we have in different parts. So for example, this is a hot spot with a specific frequency in a specific time. So we have frequency by time. These are the maps. And sometimes they just make what we call correlation with the intensity of the sound. So they also consider the intensity of the sound in terms of decibel, and they make another maps. And they can kind of what we call transformation or Fourier transformation. I will discuss about that later. And then they can see that in different frequencies, what are the shapes of these auditory evoked potentials in different, let's say, frequencies. And I told you, the reason that we have in that level of detail inside the book about auditory processing in the brain is that the first author of the book is a person who is interested in the auditory processing in the brain. Probably for those who are not going to kind of go to the details about auditory processing, that might be too much to learn about details. But at least you can have an idea about, okay, with these methods we can measure the electrical activity in the brain in different parts and measure how brain respond to different stimuli. It could be an auditory stimuli or different types of stimuli. Okay.